Thank you very much. Thank you very much, Tina, for this introduction here. And yeah, thanks also for having me here. And it's really a pleasure to be in this format in the apnoea education and talkings, because I think it's such a great opportunity to learn from each other's research and to hear from each other, also in terms of connecting each other closer. And hopefully very, very soon we can do this in presence on site again. But yeah, thanks for having this opportunity, and also good afternoon to the audience from my side. And yeah, so I jump straight into our chemistry, and today I'm happy to present you glycomimetics, how we use them as tools and as potential therapeutics. Trying to get my work done here. It does not jump, right?
No, it's still in the first slide. Now,
okay.
now it's okay.
Yeah, so hopefully this will work. Okay, so sorry about that. In the beginning, I will introduce very, very briefly Graz University of Technology to you, and also our institute, iBioSys. And then I will give you an introduction, very brief introduction also on glycomimetics. And then I will go further into chemistry, how we use immunoalditol glycomimetics as tools for activity-based protein profiling of carbohydrate-related enzymes. Then I want to talk a little bit about how we use C-glycoside type glycomimetics as therapeutics, potential therapeutics as antibacterial adhesives. And in the end, I hope I will close the circle, how we want to merge and amalgamate chemistry and technology at our institute. So Graz University of Technology, as they say, that's not my saying, Austria is in the heart of Europe. So this is little Austria here, where we are located. And Graz is towards the southern part of Austria, close to the border to Slovenia, which is very, very essential for our institute. This is Graz. And yeah, we have 3 campus locations in Graz. This is a picture of the old traditional building, the old campus, and this is the chemistry building. We are a mid-sized university, I would say, with approximately 13,700 students, around 40,000 staff. And we are one of the main partners of what has been a unique collaboration 12 years ago when we founded this, called the Navi Graz, Natural Sciences in Graz, collaborating very close in teaching and research with the University of Graz and MedUni Graz. And the strategy of our university is sustainability and internationalization. And we have 7 faculties, and I would say 96 plus 1 institute, and this plus 1 is us. Because we are a very, very young institute, we have been founded 2 years ago in 2020 on 2 happy occasions. And the first one was that Karin, and you are very well familiar with Karin Stana-Kleinschek, when she decided to leave her hometown, more or less in scientifically wise, in Slovenia, Marburg, and join TU Graz at our place. And at the same time, the second happy occasion was that somehow our rectorate thought that it is time for a professorship in glycochemistry. So this was the 2 occasions, and then Karin and I met, and we rapidly, I would say, fall in love into each other scientifically wise, because we are both interested in doing work in business as research on carbohydrates. In chemistry, we're working on small molecules, and Karin, as you all know, on large molecules. So we thought our vision was it would be pretty cool to merge, to amalgamate these 2 topics. And this is how the Institute iBioSys, the Institute of Chemistry and Technology of Bio-Based Systems, has been founded. So we are interested in the design, synthesis, and characterization of rather small molecules, glycomimetics, and produce modified biostructures. And as you all know, Karin and her group, they are interested in biomaterials and how to use organic and inorganic hybrid systems, and to use advanced processing tools for the fabrication of biopolymers and material sciences. And this is our vision. By combining our 2 research groups and research interests, we want to create advanced bio-based systems for applications, for example, in biochemistry, medicinal applications, technology applications, and we want also to work on characterization and engineering on bio-based systems. So going a little bit deeper just as an overview, we are working on the bio-based small molecules such as saccharose or even monosaccharides, and the design and synthesis. We obtain glycomimetics. We do a thoroughly characterization by NMR, X-ray, also biochemical evaluations, and then use those glycomimetics as for applications in biomedicinal science. And Karin's group, coming from the larger size scale here, they use bio-based polymers, such as cellulose, for example, other polysaccharides, hemicellulose, and with various really cool processing, such as electrospinning, spin coating, 3D printing is a very, very big topic now in the group, and by various testing in-house, and that testing really includes QCM, IR, SAX, HMF, and you name all the testing methods, and they create bio-based materials for new applications. And the vision is now having these two tools, we can design and synthesize small entities, then somehow modify polysaccharides with our small entities, which has specific biological activities. And with the processing and characterization technology available in Karin's group, we will fabricate advanced bio-based systems for technical applications and medicinal applications. So this is basically the vision we have. So we are all interested in biomass and renewable resources. And when we come to a kind of a size scale perspective, then what EPNOE members and you are all more familiar are polysaccharides, oligosaccharides. So this is biomass, for example, wood. And then the size scale is more fibers, cellulose fibers, microfibers, nanofibrils, also polysaccharides. This is your type of business. And what we do is we go one level down, coming from cellulose, for example, to glucose, and those are our starting materials. And with chemical modification, we fabricate glycomimetics for certain applications. And this is actually what I want to show to you today. So what are glycomimetics? Glycomimetics are in principle sugar analogs, so chemically modified carbohydrates. And with this chemical modification, we introduce modified properties. So ideally, we enhance the properties, the biological properties and activities we are looking for. And here are some paradigmatic examples you are all familiar with. For example, Tamiflu, shown here, is the glycomimetic of N-acetylneuramic acid. So we have a Kava cyclic here, some artificial substituents. And as you all know, Tamiflu is used for the treatment of influenza. And what it does is it's an inhibitor of the neuraminidase, and the neuraminidase is located at the outside at the glycocalyx of each cell, and is responsible to cut off neuraminic acid residues in order to cut the way free for the spread of a virus into the body. Now, if this enzyme is inhibited, the neuraminidase residues cannot get cut off anymore, and the virus cannot spread into the body. This is how it works. There's another glycomimetic miglustat, an immunosugar. It is the glycomimetic of glucose, and what it does, it's an inhibitor of glycosidases and is used for the treatment of diabetes type 2. So with this inhibitor, the enzyme is inhibited, and doing such, the blood sugar level can be controlled. And ACT is also a very well-known glycomimetic of deoxythymidine. We have an acido group here instead of the hydroxyl group, and this has been one of the first therapeutics against HI virus. And what it does is, in combination with other drugs, it inhibits the reverse transcriptase of the HI virus. And now coming to the glycomimetics our group is interested in. So we are interested in aminosugars and carbo sugars. In aminosugars, the natural occurring ring oxygen is replaced by a nitrogen atom here, and in carbo sugars by an CH2 group. And we are also interested in C-glycosyl type and C-glycosyl glycomimetics. In most of the natural occurring glycosides, we have nitrogen and oxygen linkages here, and we want to replace them by an CH2 group. In glycosyl type glycoconjugates and here in real glycosides, in order to introduce stability towards hydrolysis of those glycosides. And this is what I want to talk today about. I will start up with immunoelectrodes and how we use them as tools for studies of glycoside processing enzymes. So iminoaldehydes, as I have mentioned already, in the ring, the natural occurring oxygen is replaced by a nitrogen, and this nitrogen introduces trivalency and basicity. And they come in a large variety in nature already. A few paradigmatic examples can be seen here: 6-membered ring version, 5-membered ring version, dicyclic system, and so on and so forth. And meanwhile, a really vast number of chemically modified glycomimetics has been synthesized and known in literature. So as I mentioned already, they have a basic nitrogen, and what they do, they are very powerful, reversible in means of competitive glycosidase inhibitors. I will explain that shortly. So they come in a large structural type, and they are typically used as diagnostics and therapeutics. Now, how do they act as glycosidase inhibitors? This is shown here. In the middle, you see a natural substrate, a typical glycoside, in the active site of a glycoside processing enzyme with the catalytic acid-base residues here. And with the help of this catalytic acid residue, protonation of the glycosidic oxygen takes place, and with the assistance of the catalytic base, hydrolysis takes place of the glycoside. Now, if an aminosugar is present with the basic trivalent nitrogen here, a relatively stable bond, I would say ionic bond, can be formed between the nitrogen and the catalytic acid residue. And now by this, the aminosugar sits with this bond in the active site, and the active site is blocked for incoming natural substrate, so the enzyme is inhibited. And what is also interesting to mention here is that 5-membered ring aminosugars are typically, most of the time, even more active with a respective enzyme than a 6-membered ring. And this is because the boat-like conformation of 5-membered ring systems is closer related to the oxocarbenium ion-like transition state of the natural substrate. It is also found in a boat conformation. And the applications we want to do with aminosugars is, of course, to use them as glycosidase inhibitors, to use them as tools for mechanistic investigations, as therapeutics for pharmacological chaperone therapy. And today I want to talk how we use them as tools for activity-based protein profiling. But first of all, before we can apply them, we actually have to synthesize them. And since many years, we are working on the design of really concise and efficient synthetic methods towards such compounds. And these are two methods we developed for the 6-membered ring version. So typically we start from 5-azido-aldoses, and we have really a good number of different configurations here, very easily available. If we want to introduce a modification at the position C1 here, the pseudo-anomeric position in aminosugars, we take advantage of the Staudinger-Azavitic nucleophile reaction sequence, where we first reduce the A site to the amine, which can then close the ring with this protected aldehyde substituent here to an iminium ion, and with a nucleophile present, this iminium ion can undergo attack at this position here, and we end up, depending on the nucleophile, with different modifications here at the position C1. If we want to do the modification at the ring nitrogen, we just do a very simple intramolecular reductive amination. The A site again gets reduced to the amine, which can then attack the aldehyde, and under reducing conditions, we obtain the aminoaldehyde, which can now undergo chemoselectively reactions here at the ring nitrogen. And we really have also a very nice toolbox here in different substituents and spaces and so on, what we can introduce here. When we want to go for the 5-membered ring version, we have also two nice approaches here. If we want, again, to introduce the modification at the position C1 here, we take advantage of the Amador arrangement. This is a pretty cool reaction, and I'm going to explain it later on in my second part. So what happens is, in one step, you introduce an amine at position C1 under concomitant isomerization of the aldose to the ketose. Then again, ring closure in an intramolecular reductive amination gives you the 5-membered aminosugar already with the amino substituent here at position C1. And follow-up chemistry can modify this 1-amino-deoxyaminosugar either at position C1 here or at the ring nitrogen, also read you selectively. If we just want to modify the ring nitrogen, we take advantage of an enzyme here, and this enzyme is a glucose isomerase, and this enzyme is one of the the largest scale enzyme used worldwide, because the soft industry uses it for the isomerization of glucose to fructose for soft drinks, because fructose is sweeter than glucose. So what this enzyme does, it also isomerizes aldoses to ketoses. If we then perform the intermolecular reductive amination, we obtain a 5-membered ring iminoaldehyde and do chemoselective modification here during nitrogen. And over all these years, we have developed a very, very large structure activity study here. So we don't know exactly, but I should say we know very well which substituent we have to place here in order to fine-tune the biological activity. And we have now a range available from the micromolar scale to really down to the single figure nanomolar scale. So we also have very, very potent inhibitors, just depending on what we introduce here. And this brings me to the application I want to show you today. So we use immunosugars as tools for a technology called activity-based protein profiling. And this is really a very, very versatile tool in order and technique to study enzymes. And what those probes do, they do not really detect only the abundance of enzymes, but their real activity. And this is the big difference. So you can look in really complex environment in cell lysates or cell cultures or living organisms for the really effective activity of enzymes. And with this technique, you can, for example, identify protein function. You can determine up and down regulation of enzymatic activity. You can do disease diagnostic. You can look and discover new ligands, inhibitors, therapeutics, or you can even identify completely novel protein targets. So very, very versatile and very important technique, which has been developed during the last, I would say, 20 years and is now really available at hand for many different classes of enzymes, such as lipases, kinases, phosphatases, methyltransferases, and also for glycosidases. And the principle is shown here. So typically, when we want to investigate a proteome, and here in one specific enzyme here, we have a probe, and the probe consists of two very important ingredients, I should say, or parts. One is the warhead. The warhead is a specific ligand for the protein of interest and also contains an electrophile, which allows covalent binding to the active site of the protein of interest. And then we have a tag, a fluorescent dye or enrichment tag for visualization and quantification, typically done by MS or SDS page. And this would be a one labeling strategy, and there's had also been introduced a two-step labeling strategy. So here the probe looks a little bit differently. We again have the warhead, selective electrophile for a certain protein, and we have here ligation handle, for example, an A site or alkyne, which can then, in a second step after labeling of the protein of interest, undergo the tagging with the tag. Sometimes that might be of advantage for different enzymes. And in terms of glycosidases, really very, very important contributions have been made by Vocadlo, Bertozzi, the Witters group, and Overkleft now has developed against really a large group of different glycoside processing enzymes, really powerful tools. Yeah, so we were also interested in doing this kind of techniques. However, we cannot really compete with those big names and those big groups. So we thought we try to be complementary in a technique. And this is how we got interested in the ligand-directed protein profiling. This principle was introduced by Hamashi to all different kinds of enzymes. And the mechanism is a little bit different, and the probe is designed a little bit differently. So we again have a ligand here, which is selective for enzyme of interest. However, this ligand now is a reversible inhibitor. So we don't have an electrophile present, so it's a competitive inhibitor. And then we have again the tag hemisphere of the probe, which contains either a fluorophore reporter group or a ligation handle. And now we have a third compartment here, and this is a reactive group. And this reactive group is placed in the middle via a linker here. And this reactive group can be nucleophilically attacked by an amino acid, which is in proximity of the active site, not located in the active site, but in proximity. So how it works is the ligand gets recognized by the enzyme, interaction takes place, and this reactive group now allows for being nucleophilically attacked by an amino acid available at the entrance or in proximity to the active site. It reacts. Very crucial here is this spacer length, because this spacer length brings the reactive group into the proximity here. And this reaction, during this reaction, the tag gets covalently labeled to the enzyme. However, the ligand is set free. And now, because we have a competitive inhibitor, it can be released of the active site, making place for incoming natural substrate. And this is the big difference. So in this technique, the labeled enzyme remains its activity. And this is what we wanted to show for glycosidases. Following the probe design of Hamashi, we translated to our sugar probes here. For the ligand, as I have mentioned already, we take advantage of immunosugars as they are ligands, they are inhibitors, and they are competitive, reversible. For the tags, we use fluorophores or ligation handles for a two-step labeling, which we prefer at the moment. And then we have this crucial reactive group. And Hamashi introduced ester groups, sulfonates, acyl iminosols, and activated ester. And it's kind of a playing around here in this region, because esters, you know, they are not so reactive. However, if they are not so reactive, the probe itself is rather stable. And activated esters here, they are very, very reactive towards nucleophilic attack, but that also makes the probe itself very reactive and unstable. So what you gain in reactivity, you lose in stability. This is part of the game what you have to play. And today I want to show you our work on sulfonates here. So first of all, we had to do the math. This is, first of all, choosing model substrates or model targets, and we went for beta-glucosidases. In that particular case, we did work on beta-glucosidase from Dermatoga maritima. And first of all, we had to look whether there is a suitable, for our probe, suitable amino acid available at the entrance of the active site. This work was done in collaboration with Karl Gruber from the university. So we modeled a probe we have designed into the active site with the amino sugar sitting there in the active site and having the handle hanging out there towards the entrance with the sulfonate group here. And we were looking for amino acids and indeed found 4 suspicious suspects, I should say, a tyrosine, histidine, and 2 glutamates, which are in proximity here located and could react in principle with the tosylate here. And this is how also we designed the length of the spacer here or the linker here between the ligand and the reactive group. Okay, so this one should work in principle. This was fine. Next, we had to do the synthesis. Again, synthesis we designed straightforwardly, I would say, starting from this benzoic acid sulfoneuclide. Esterification takes place with this acidohexanol, leading to the desired sulfonic ester. Now having here an A site at the hemisphere of the tagging of the probe. In order to install the other handle or the probe on the probe hemisphere, we take advantage of a very simple amide coupling, this aminohexanol or aminododecanol, in order to having 2 different spacer lengths here under mixed anhydride, classical mixed anhydride conditions to install the amide bond. The alcohol can simply be oxidized by this Martin reagent to the aldehyde, and the aldehyde can undergo substitution reaction with the ring nitrogen, leading to these 2 probes here with 2 different spacer lengths between the reactive group and the ligand. And this is how the labeling actually should happen. So this is our probe with a spacer bringing the sulfate, the sulfonic acid here in proximity to the amino acid. And this would be the enzyme, for example, with a tyrosine, and the alcohol should be able to attack nucleophilically here at the tosylate. In this reaction, the tag, the A site, gets covalently bound to the enzyme and the ligand is released. And then in a second step, we use a click kit, a commercially available click kit, in order by and click chemistry to put the fluorophore on in order to be able to measure then our labeling and to quantify our labeling. And what is also crucial from the interaction kind of respect is Here we need a rather strong inhibitor in order that the selective interaction between the enzyme and between the target enzyme and the probe can take place. And now when we release the ligand or want to release the ligand in order to regain activity, this should be a less active inhibitor. On paper, in principle, would be cool if we would found that. And indeed, next we investigated the Ki values, the ligand potency against the two enzymes we have chosen here as a model. I've mentioned already beta-glucosidase from Dermatoga and also beta-glucosidase from almonds. Here are the Ki values. This is the parent compound, 1-deoxynitromycin. This is the shorter probe. This is the longer probe. And this is the what we call it truncated version, the ligand, which should be able to leave the active site again. Looking at those numbers here, just roughly, our compounds are more potent. Looking at Dermatoga, more potent than the parent compound. This is good news. They are in the probes are in a range where they should be able to work as probes in the high figured nanomolar range. So we have a Ki value of 550 nanomolar for the shorter version, 144 nanomolar for the longer version. And luckily, it turned out that the truncated version here has less potency for the target enzyme with 93 micromolar. So at least two numbers of magnitude less active than the probe itself. So this is brilliant. This does brilliantly, perfectly fit in our ideas. And for the almonds, we got kind of the same picture, not as good the difference here between the probe and the truncated version, but still numbers we could work on. Now, next was the labeling experiment. And I would just show you one preliminary result we got here with the beta-glucosidase from almonds with the shorter probe. We did kind of a time-dependent study here in labeling. What you see is the SDS page here. Here we have the ladder in order to see whether we have the enzyme in its active form with the right mass. And then we incubated the enzyme 5 hours, 10 hours, 15 hours, and 20 hours with our probe. Did the click kit addition of the fluorophore, and then went to the SDS page. And we see here in the fluorescent readout a textbook labeling, I would say, of the enzyme in the time dependence manner. So 5 hours in the short incubation, we see hardly any fluorescent. And then with increasing time, we see also increase of labeling. So this is really, we were very, very happy. And this is the result I wanted to show you here. So that's the first time that the ligand approach was applied successfully on glycosidases. And there's, of course, you can imagine further work going on. Now, I want to switch gears to the second part of my talk. And now I want to introduce C-glycosyl-type glycoconjugates and how we use them now as potential therapeutics, I should say. And here is, for the synthesis, I've mentioned already that we take advantage of this really cool Amador arrangement. It's cool in two senses for my taste. It's a very well-known rearrangement. Already our all-sweet grandfather, Emil Fischer, has been working on that direction. And what happens here is it's the reaction of an amine under acidic condition with an aldose. In one reaction, an introduction of the amino substituent at the position C1 takes place. And under acidic conditions, the isomerization from the aldose to the ketose takes place. All together in one reaction without the need for protecting group manipulation. So this is really cool in carbohydrate chemistry. And we end up with this C-glycosyl-type conjugates. And what we wanted to investigate here together with Professor Lindhorst in Kiel, we wanted to show if such compounds could inhibit bacterial adhesion processes. So how does that work? The bacterial infection typically has a bacterial adhesion process beforehand, and how that works. This is an E. coli, for example, and at the glycocalyx, so at the outside of the bacteria, we have protein tentacles here. Pilys are very, very fine protein chains. And at one of those protein chains called type 1 fimbriae, at the tip of those proteins, there's a carbohydrate recognition domain called a lectin, also FimH in that particular case, which has a specificity for mannose residues. And what the bacteria does when they want to interact with the host cell on its glycocalyx always presents sugar, glycoproteins and sugar chains. And we have also mannose residues sitting here. So the lectins here recognize and interact with the mannose residues presented at the outside of the host cell. This is the adhesion process, and the adhesion process initiates the bacterial infection. Now, if an artificial ligand, mannose ligand, is present here, the carbohydrate recognition domain of FIMH gets blocked by these mannose residues. And because it's blocked now, it cannot interact with the cell anymore. The adhesion event is inhibited, and bacterial infection cannot take place anymore. And maybe you're familiar that mannose itself is on the market already in combination against a specific bacterial infection with antibiotics, especially in the urinary tract infections. This is used. However, mannosides are better inhibitors or inhibitors for this process, and even substituted mannosides are even better inhibitors for this process. If we compare, for example, this metal mannoside aspirin compound with a relative inhibitory potency of 1, the Lindhorst lab introduced compounds here with 70-fold increase in activity, or even 7,000-fold increase in activity against this process. So very, very potent compounds. And as we all know, also that lectins, they prefer a multivalent presentation of the ligand. And this was what we wanted to show also with another cooperation, Ludovico Landemare in France, and together with Tispe Lindhorst. And in order to have a multivalent core, which is bio-based, we decided to go for BSA as a multivalent core. And we wanted to click on our Amador rearrangement product. So we had to synthesize first the mannoside ligand here, straightforward Amador rearrangement of this aldose with this acidoethylene glycol amine. Rearrangement takes place, and we end up with this compound. We have a mannoside residue here, and here a ligation handle. And next we have to modify the BSA with this succinylbutyl activated alkyne compound here. We clicked, we did an amide bond formation. This is how we introduced the alkyne motifs on BSA. And we obtained a multimer where we found 39 alkynes here. And then in a click chemistry, we just simply click on the A side to the alkyne here in order to have the mannoside presented on BSA as a multivalent core. We obtained two main products, one presenting 15 mannoside residues and one 23. Then we went into biology evaluations. We tested here different types of lectins, which are all mannoselective. We have 3 plant lectins, 1 bacterial lectin, and 2 recombinant human lectins. This again is the parent compound, the methyl mannoside. And this is another monomer we investigated. And here in this lane, we see the multimers, the multivalent glycoconjugates we have been synthesized. And we see that very nicely for some certain lectins, we obtained up to 2,000 time increased potency for this lectin compared to the parent compound. So those are really, really nice results, and we were very happy with this. And now it comes together with technology. So we thought if BSA is a very good multivalent core, and here we started a collaboration with the Heinze group at the University of Jena, why we don't, or can we use polysaccharides as multivalent core, as there is also polyglycomers available as such FIMH inhibitors. And we thought we just, in a proof of concept, try if we can modify alkyne modified cellulose in a click chemistry with acido sugars available from our group. So we chucked them together, and indeed we found that in all cases, we got very nice modification after the click chemistry of the modified cellulose with the respective sugar, small sugar entity. So what we have available, and we did it also the other way around, I should mention that we also successfully clicked on probachyle or alkyne modified small sugar entities to acido modified cellulose in the same process. So what we have done in this proof of concept, we found a very nice way via a click chemistry approach to modify polysaccharides with small sugar entities. And this opens up the opportunity to introduce now tailored properties via those small sugar entities to the polysaccharide scaffold. And that would mean that we are able to synthesize or to fabricate artificially branched polysaccharides for selected applications depending on what the sugar entities are here. So together now with Tispe Lindhorst, looking again into FIM-H activity, we synthesized straightforward this proper-child monocyte here, got the triple bond in a O-glycosidic fashion on the monocyte, and in a click chemistry on a cytomodified cellulose, we clicked on the monocyte via the triazole to the polysaccharide backbone. And just hottest from the Tispe Lindhorst lab, we last week got the first results, not in numbers, unfortunately, yet. However, in the very first experiments, bacteria really do adhere onto the cellulose backbone with the monocyte residue. So this is really, really exciting, and I can't wait for getting the numbers and taking synthesizing more of those compounds. And we want to look into real C glycosides as well here in order to get this connection here, the glycosidic linkage, more stable. However, with this process, we have done the proof of concept. Now we open up a really huge playground, again, in the collaboration with the Heinze lab. So what we really have in hands now, and what we are going to look into it, we can, for example, from the polysaccharide perspective, we can use different polysaccharides as backbones, cellulose, dextran, xylene, pectin, you name it, you are the experts in this field. We can also use different ligation methods, so not just simple kappa assisted click chemistry, but we can also look into other ligation reactions here. For example, norborane and so on and so on. There is a vast number of orthogonal ligation reactions available. We can introduce different spacer lengths and also different types of spacers. For example, alkyl spacers, ethylene glycol spacers, and so on. And also we can have different degrees of substitutions here at the polysaccharide side. So really a large playground on the polysaccharide side, and also on the small entities, glycomimetics here. With monocytes, we introduce FIM-H activity. With other configurations or other substitution patterns here, even hetero substitutions, we can introduce polyvalency, different physical and target properties. For example, aminosugars can be introduced in order to obtain multivalent glycosidase inhibitors, and so on and so forth. So it's really, I can't wait touching and going further deeply into this project here. And this now closes the circle, how we would have a tool or have a tool in hands now for our vision to merge, to amalgate the chemistry and the technology at iBioSys. Because having said that we can fairly effectively synthesize biological active small sugar glycomimetics entities here. We have in a proof of concept study now methods to conjugate, covalently conjugate those entities with a specific biological activity onto polysaccharide backbones here. And now what we obtain is polysaccharides with certain biological activity here. And what we can do with this material now enter the technology group to use these cool processing methods they have in order to fabricate new bio-based systems for their cool technology and applications, which is going on already in Karin's group, tissue engineering, material investigations, and 3D print. And last but not least, one topic we are also interested in to build up together at our institute iBioSys is the modification of surfaces. So in order to modify for biomedical applications. And here we all know that the modification has to be biocompatible. And what can be done on modification of surfaces? We can introduce, for example, or one can introduce, for example, resistancy against biological foulants, resistancy against bacterial adhesion, or we can modify in a term that we have absorption of molecules or adhesion of cells. Even we can release molecules, release drugs, even employ chemistry here. So there's a lot of different modifications possible here. And what has been done in Karin's group already is pretty cool also, I found. What they have done is lately on a cellulose film, they introduced an activation group here in imidazole activation of the primary hydroxy here. And then they could also kind of click on or react on lysine amino acid doing a carbamate bond here. And this film actually show very nice biological and very interesting biological activities. So it significantly decreased, for example, the adsorption of BSA and other fibrogens. And that would mean that such modification on surfaces are very, very interesting for a potential application in, for example, anti-fouling, to create anti-fouling surfaces on dishes contacting blood, for example. And another cool thing was done in her group by a very talented master student, Tanja Katan, who managed by spin coating to produce chitosan films, and via typical amide coupling reaction conditions, she could couple on two amino acids, two glycine amino acids here onto the chitosan film. And this is really, really, really cool, I found. So actually she did peptide synthesis on the surface of the film. And in this paper here, which came out recently, she did also a thoroughly characterization of her surface. And this is how we want to bring chemistry into it. We want now on such surfaces, not directly on chitosan films yet, but on amino modified surfaces, bring on and couple on the Amador arrangement product, where we have a sugar molecule and also a lysine residue here. So what we want to do is we want to click on via an amide coupling here our glycoconjugate, then could get rid of the Bock group here and extend with further peptide coupling here the glycopeptide on surface. And those are just two compounds we also want to investigate. So this is what I wanted to tell you, what's going on from the chemistry side in our institute today. So having this said, I told you and showed you that we have a very broad structural diversity of immunosugar type glycomimetics available in our group, which we use as tools for activity-based protein profiling. I did not talk about pharmacologic chaperones today, which is also going on in our group. And also did talk about C-glycosol type multivalent antibacterial adhesives. And now bringing chemistry and technology together, this with the modification method, we have the proof of concept now available to conjugate small molecules onto a polysaccharide backbone. We could develop really tailored functional polysaccharides for different technology. So it's very, very exciting what's going on in our group. And I just can tell you, stay tuned. There's more to come. With this, I want to thank all the people, and this is an amazing bunch of people, I have to say, now working our institute, and what they have done during the last 2 years to establish the institute is just amazing. And I'm not talking about the two golden girls here in front, but I'm talking about the technology group and the chemistry group. They are fantastic, do fantastic work. Thanks for that. I showed you from the collaborations today, chemistry and projects with together with Thomas Heinze, University of Jena, Ludovico in France, and Tis Belintost here in Kiel, and also Steven Withers, of course. I showed you collaboration with Karl Gruber doing the molecular modeling on some compounds. The funding comes from the FWF. This could be more we are struggling here, but we want also be successful here. And now finally, I want to thank you for your kind attention. Thank you.